For the last 20 years or so, the Varroa mite has been barely under control. The latest in the sequence of control attempts makes use of RNA interference, RNAi for short, a molecular approach radically different from all previous methods. Recently, the first commercially approved product using RNAi came to market. The question, of course, is should you use it? In this video, we'll review the first scientific study conducted in the US to look at the effectiveness of this new product called Norroa. I'll start by explaining what RNAi is, and I'll conclude with a few words about future prospects. In short, although the initial results are promising, I think we can expect much better in the future. First, let's start by explaining what RNAi is. It's not a traditional pesticide, that is a chemical that is poisonous. Rather, RNAi is a basic molecular biology mechanism used by cells to turn down or off genes. A cell needs to be able to control the use of its genes, and it has many ways of doing it. RNAi is one. We don't need to get too much into the biological details because it's complicated, but I'll say a few words about it for those who are interested. DNA stores information permanently, but when that information is needed to make a protein, for example, a temporary copy of the DNA is made. That copy is called a messenger RNA, mRNA for short. Basically, a double-stranded RNA molecule, which I'll just call an RNAi construct, which is the active ingredient in Norroa, for example, targets a single-stranded mRNA molecule that has the same sequence that it does, and that turns down or off the gene with the same sequence. That's really all the biology we need. RNAi has the best potential to control pests of all kinds relative to other technology for three reasons. First, any gene can be targeted for disruption. Many serious diseases, as I'm sure you know, are caused by genetic problems. RNAi allows us to create as many genetic problems as we like in the target pest, including many that should be fatal. Second, because a genetic sequence is being targeted, there can be nearly no off-target effects when done right. Even if the bee, for example, has the same gene that is targeted in the pest, the Varroa mite, the product won't hurt the bees because the bee's genetic sequence for that gene is so different from the mite's. Third, and finally, it's difficult to evolve real resistance to RNAi because new genes can always be targeted, or the same gene can be targeted with a different RNAi construct. Let's now talk about practical work on RNAi and pest management. First, RNAi isn't just about bees and mites. The big problem in pest management, of course, is control of insects on farms. Controlling bugs that eat our food is huge, and you would expect much research on RNAi in that situation if what I've been saying is true, and there is a lot of research. In short, there's been a lot of success on a small scale. It's been proven that RNAi can control pests on many crops. In principle, people are excited about the potential of RNAi to control pests on crops. There are also a few RNAi treatments that are already in large-scale use, but overall we're not quite there yet in terms of RNAi playing a major role in agriculture. With bees, we're basically a little behind where we are for insect pest management. Early work showed you could disrupt mite reproduction and survivorship using RNAi in the lab. This early work used injections and other methods that show that RNAi can kill Varroa, but which could not work in a real hive. A breakthrough happened when it was shown that mites floating in a solution containing the RNAi construct would take it up and be hurt by it. This led to research that ultimately led to the subject of this video, Norroa. In Norroa, the RNAi construct targets a gene important for mite reproduction, and is added to a sucrose solution which is fed to the bees. The idea is that after the hive takes it up, the nurse bees will add the construct to the brood food. The Varroa mites should then float for a period of time in the brood food and take up the construct. This should hurt their ability to reproduce. The first US study testing the effectiveness of Norroa in real colonies came out a few months ago. They did a simple experiment. Treatment hives were treated with Norroa, while controls got solution minus the active ingredient. They then measured mite levels for a few months post-treatment. They did the experiment twice, once during a dearth and once during a nectar flow. All the work was done in Florida. First of all, they found that the product basically works as advertised. The bees took up the syrup and it did get into the mites. They also showed decreased reproductive ability in the mites. Now let's consider the real question, which is whether the mites were controlled. First, they did not find any control during the dearth period. Why wasn't clear, but this suggests that the product might not work under all circumstances. In the experiment run during the nectar flow, they did find that Norroa stopped the mite population from growing. The results are shown in this figure. There was an initial drop in mite numbers after the treatment, but the mites recovered after about 6 weeks to the pre-treatment levels. Mite numbers then stayed there until the end of the experiment. The most favorable interpretation would be that the product decreased the growth rate just enough to hold the mite population steady, at least for 3 months. Of course, this is a small-scale study, about 36 hives over a few months. We need to see what happens for at least 2 years with a much larger number of hives to really be sure about these results. But they're promising. If Norroa really does stop mite growth, that would be great. However, we have to remember that they found no effect during the dearth. So if the effect they found in the nectar flow is temporary, then the next treatment could fail and the mites might come back. Of course, this is speculation, and one could speculate forever about what might happen in other circumstances. Only more data will tell. Also, and just as important, it's hard to know how effective this control is compared to other treatments. In future studies, they should consider running larger trials in which they have controls, Norroa, oxalic acid, and amitraz, for example, treatments all going on at the same time and place. This would allow us to see both whether Norroa works and how well it works compared to the other currently used products. So what can we conclude from this study? Basically, the results are promising, but we need more data to put it into perspective relative to our other options for controlling mites. Second, the product is pretty expensive, and this could limit sales. But I personally hope not. I hope it does well. Because the real take-home message from the study is that RNAi can work in real honeybee colonies in a mass-produced product. You might say it didn't live up to its potential, but you would be mistaken. If you had asked me beforehand, for example, if I thought this product would completely control the mites, like Apistan did before the mites evolved resistance, I would have said no. Not because RNAi can't do the job, but because this product only targets one gene. I suspect that you have to target many genes at once to get very strong control. In general, going from proof of concept to final product always takes a long time, no matter how much better the new solution is. LED lights, for example, were discovered decades before they took over from filament bulbs. But once they passed the filament bulbs, there was no looking back because LED lights are just fundamentally better. I believe it will be the same with RNAi versus nasty chemicals. Here we're pitting molecular biology versus chemistry, and I think molecular biology will ultimately win. As more successes roll in and more research gets done, the cost of RNAi will go down and the effects will get better. In 20 or 30 years, we might even forget how much drama there was over old-school pesticides. Well, that's about it. I started by asking, should you buy it? I think the answer is first, can you afford it? And second, are you willing to risk losing a few colonies if it doesn't work as advertised in your area? For me, I'm going to give it a shot here at UC Davis with about 10 hives. We'll see how it goes. If you found this content interesting and useful, please like and subscribe. It would really help me grow the channel. See you next time.